Okay, am I good here?
Yep, absolutely. Go ahead.
All right, thank you, Jeff. And I would like to say thank you to you and the whole team of people that put this meeting together. I mean, I know we all wanted to be in Dallas face to face, but I think for a virtual meeting, this has gone really well. And I know it was a lot of work. And so thanks to all of y'all. And in particular, I've really enjoyed the YouTube poster feature. And hopefully that might be something that we want to keep even after we come back together in person. But for the next few minutes, I want to talk about the release of peanut germplasm lines with resistance to late leaf spot. There were a lot of people that made significant contributions to this work. So these are all the co-authors on my presentation. Most of these names are very familiar to y'all, to this audience, but there's a couple that you know, some of you may not have met. One is Ali Lamone. Ali was a graduate student, got his master's degree under the direction of Peggy Akins. And we're going to be talking about some of his data. And then Ignacio Godoy is a peanut breeder in Brazil. And I've actually known Ignacio since he was a graduate student at the University of Florida under the direction of Alan Norden. So my talk's going to be split into two different parts. The first part we're going to talk about is the release of TifGP-3 and TifGP-4. And then we'll finish off, the last part will be the release of TifGP-5 and TifGP-6. So to start off with TifGP-3 and 4, a population was developed by crossing this Brazilian breeding line with TifNV-High O. And as we've heard in a number of different talks this week, this Brazilian breeding line is one of the ones that had the 3 introgressions from cardenasii, 2 introgressions on chromosome AO2 and 1 on chromosome 3. So we made F1 we created F1s and then made an F2 population, which were then genotyped. And we selected some individuals that were homozygous for all 3 introgressions. And then we selected other combinations of these introgressions. We advanced this down and the red part is where Ali came in. He did an in vitro detached leaf study with some of this material. And then, he also did field studies at 2 locations, replicated field studies at 2 locations in 2018. Seed was limited, so these were small plots. It was replicated, but it was small plots. So we wanted to follow up after he left, and we did an additional field study with larger plots, and we'll present data from all those studies. So this is just a diagram of the genetic material that Ali had available. He had some individuals from that cross that had all 3 segments. And then he had individuals that had just individual introgressions. Like this is an individual that was homozygous for the introgression on AO2. This is homozygous for the introgression on AO2, the bottom of AO2. And this is individuals homozygous for the introgression on AO3. He also had all the pairwise combinations. And of course, we had a selection from that cross that did not have any introgressions. I'm just going to hit the highlights of Ali's study. It was actually published earlier this year in Crop Science if you're interested in more details. But this is some of his detached leaf data. And to begin with, of course, we've got the susceptible checks that had a large number of late leaf spot lesions and sporulating lesion progression. And then the highlight is this yellow line is the Brazilian line. Then we have the green line, which is the individuals that had all 3 introgressed segments. And this red line is individuals that had just the introgressions from the top of AO2 and the bottom of AO3. So we see that those are the best lines on both of these graphs. And another significant point here is that there was really no significant difference between the individual that had all three introgressions and the individual that just had the two introgressions, one from AO2 top and the other from AO3 bottom. As I mentioned, seed was limited, but he did have replicated small plot studies at two different farms in 2018. One was the Gibbs farm, the other was the Lang-Rigdon farm. These were non-sprayed trials, the same type of genotypes that we just talked about were included in this study, along with some checks. This is a picture of just the range of leaf spot resistance that we have in this material. This is TifNV-High O. This is the Brazilian breeding line. This is one of the F2 individual, or traces to an F2 individual that did not have any of the introgressed regions. This is an individual that had the two introgressed regions, one from AO2 top and one from AO3 bottom. And this is an individual that had all three introgressions. So looking at the some field data from the Gibbs farm, of course, this is leaf spot severity, Florida scale, and you know, the susceptible checks are eaten up with leaf spot. But the three lines that we highlighted in the in vitro also were the best lines for leaf spot severity, the Brazilian line, the all segments, and the AO2 top and AO3 bottom. Same thing with AUDPC. And similar results at the Lang-Rigdon farm, susceptible checks, the best three lines. And once again, an important part is there was no significant difference between the individual that had all three and the individual that had AO2 top and AO3 bottom. Yield, the Brazilian line is not adapted to South Georgia, but TifNV-High O is. But you can see that we've, even just with a single cross, we've made pretty good progress in coming back to the yield level of TifNV-High O. Pod weight, the Brazilian line has small pods and seeds, but we've come back towards TifNV-High O. And the conclusion from Samueli's study was that major genes for resistance are contained on the introgression from the bottom of AO3 and the top of AO2, and that the introgressed region from the bottom of AO2 did not add significantly to the level of resistance to late leaf spot. As I mentioned, we followed this up the next year with some larger plot research, and this is a good shot of showing you, the variability that we have in the level of resistance here. There's a lot of stuff on this slide. You can ignore the top half. Juliet at some point will talk more about this. This is some induced allotetraploids and some crosses with that material, which of course has near immunity to high levels of resistance. These are the susceptible checks, Gregory runner 886, flow runner, and the line that had none of the introgressions. Very high level of leaf spot severity. And then we've got these two germplasm lines, TifGP-3 and TifGP-4, that have a level of resistance very, very similar to the level in the Brazilian line. So we're releasing two lines. TifGP-3 has all three segments. TifGP-4 has just the two that, and we're releasing the one that has all three, just in case something comes about that somebody needs that third introgression. I think really most breeders would want to use this because there's less likelihood of linkage drag. So a question is certainly in probably some of your minds, what about nematode resistance in high OL? Because TifNV-High O is nematode resistance in high OL. Well, we didn't select for that. So what's available right now is segregating for those traits. However, Juliet came to me in the spring and said, you you genotype some of these seeds, and we have information now on nematode resistance and high OL. So do you want these seed? And I said, sure, I want these seed. And so we're increasing them this year. So as a bonus for sitting through my talk, if you want to have some seed with these introgressions, and you also want information on nematode resistance or high OL, get with me in the winter and I can share a small amount of seed with you. The material that we're releasing is segregating for these traits. Moving on to the next group of genotypes, it's TifGP-5 and 6, and this traces back to the genomics project and the one of the KASP populations that we developed with Tom Islib. This is the C1801 population. Juliet has taken the lead on this. She wrote this up. It's not yet published, but she's taken the lead on this. But the population was a Florida 07 by GPNCWS16 population. We made the cross, we advanced the F1s in Puerto Rico, and then sent half of the F2 seed to North Carolina. We kept half of the F2 seed, and then advanced that to the F6, where we harvested individual plants and increased seed for development of a recombinant inbred line population. So we had 193 in the Georgia part. Tom had 192 in the North Carolina part. We used this part to phenotype for leaf spot and do QTL mapping. And then they identified 3 QTLs, and we validated those markers using the North Carolina half of the population. And all that's been published. That's published information. So we're going to release these 2 lines from that population. And just a little bit of data. I'm almost finished here, Jeff. Leaf spot, you can see it was phenotyped for several different years. This is the average of 4 years. These 2 lines that we're going to release ranked 2nd and 3rd over all the recombinant inbred line population, and that compares very favorably to the germplasm line that came from Tallery and Tom Stalker. And then once again, we included this, or Juliet did, in this big data, big test that she had. And here's TifGP-5 and TifGP-6. So levels of resistance comparable to the Brazilian line and the 2 other germplasm lines that we're releasing. Why are we releasing 2? Well, TifGP-5 is high oleic, TifGP-6 is normal oleic. And like I said, there's markers. The interesting thing about this slide is one of the QTLs for resistance actually came from Florida 07. The other 2 came from the North Carolina germplasm line. Last slide, yield of this material is very good. I mean, you can see here's the 2 germplasm lines up here, and then here's Florida 07. And these lines have yield comparable to Florida 07. So really, actually, a few years ago, I was considering releasing these as cultivars or a cultivar, but the grade needs improvement. So that's why we didn't release it as a cultivar, but it does have excellent yield potential along with the leaf spot resistance. So we're releasing 2. QTLs are available. If you want to use these in your breeding program, you'll have markers that you can also use. And those are the funding sources. And yes, Jeff, I can hang around for questions for as long as people want to ask them.
Okay, great. I think the next session starts at 11:30. So there were a couple of questions. I'm going to start back. Alicia is still on, I hope. I think Sham had a question for you, which was, what were the resistant accessions of A. cardenasii that you identified?
Can you hear me? Yes.
Yep.
Okay, so we have more than one. I can refer to him because there are several candidates, and I show a few of them, but we, like I mentioned in previous meetings, we are using the 1017, 1000. That is the one that is a reference, and some previous work as kind of as a reference from previous work, but we haven't identified new ones, and we can, yeah, I can talk with him about that. But
Okay.
there are several ones. Yeah.
Okay, thank you, Alicia.
Yeah.
And then we have a question from Barry Tillman. So I guess this is directed towards Corley. What would be the practical impact of fungicide applications from the AO2 and AO3 introgressions? Could we reduce by half? And that, I think that's a great question.
Yeah, and so we are trying to get a handle on that, Barry. We did a study last year looking at different fungicide applications. And we were going to put some economics to it and try to answer that question for the growers. Unfortunately, our full fungicide spray test was in a field that just was not a good field. And so our full fungicide sprays yielded less than our no fungicide sprays. So we really couldn't do any economics. And it was just because it was in a bad field. It wasn't, you know, but I really, we're looking at two different things. One, no fungicide sprays, or the second option would be just an application of convoy to control white mold. That's what I'm hoping for.
Interesting. And then I guess follow up to that is from Charles Chen. I wonder if this is additive effects for the 3 genes.
I don't know the answer to that, Charles.
Okay. And I guess the last question from Bryce, and I can answer this one. I guess this is in terms of you know, the leaf spot ground cover. So if you were to have significant time between the pre and post leaf spot flights, what is the method to differentiate between leaf spot effect resulting in decreased percent greenness versus shoot growth increased percent greenness? So basically what we did, we took a very naive approach to that, and we just looked at what is the difference between that first and second flight, even if there was, you know, a month apart. We do have bi-weekly flights that we can go back and do like an AUDPC type calculation, but you know, for the most part, our final flight was what we essentially used. And if it was negative, we just used it. You know, if it was a continuous variable, we just used it. So same question for plant density is more important. Upper layer could be impacted by leaf spot. So usually leaf spot is just, you know, on the upper area, and you start to notice the defoliation on the underneath of the canopy. So Albert Colbreth has a comment. Most of the evaluations have been where the individual genotypes were exposed to inoculum for other plots, so full field production may be even better for leaf spot management.
Yeah, that's a good point, Albert.
Yeah. So that concludes our session.